Welcome to another Microbiology Nerd video. Today we'll be exploring some of the essential techniques used in the lab to help us identify microorganisms. Mastering these techniques is crucial for accurate diagnosis and treatment of infectious diseases. Just as a side note, the methods discussed in my videos are typically practiced in UK labs, so I acknowledge that variations may exist due to regional regulations or practices. Without further delay, let's dive in. Sterilisation and disinfection. Sterilisation is the process of complete elimination of all microorganisms, including spores. Sterilisation is achieved through methods such as dry heat, filtration, and most commonly in lab, autoclaving. Autoclaving at high temperatures helps destroy bacterial, fungal, and viral contaminants. Now, this is slightly different to disinfection. While sterilisation eliminates all microorganisms, disinfection only reduces the number of pathogens to a safe level for handling. Disinfection methods include using chemical agents like alcohol, bleach, or UV radiation. In the lab, it is considered good laboratory practice to wipe surfaces before and after testing patient samples. This ensures the overall quality and reliability of test results. So why are these techniques so important in the lab? There are several reasons. They prevent contamination. Contamination can lead to inaccurate results and misdiagnosis. By sterilizing equipment and maintaining a clean environment, it gives us confidence that our patient results are valid. They protect staff from potentially pathogenic microorganisms. This is vital for maintaining a safe working environment as it prevents accidental exposure and illness. They also preserve valuable samples. Lumbar punctures performed to obtain CSF samples are invasive procedures, and having to repeat them due to a sample contamination can lead to increased risk of infection, as well as forcing the patient to re-experience the pain and discomfort caused by the procedure. Although rare, even more seriously, it can lead to potential complications like headaches and bleeding. Maintain testing integrity. This prevents false positive results, and it helps us comply with regulations set by government agencies. Fun fact, did you know that the Egyptians were one of the earliest civilizations to practice sterilization techniques? They used hot sand, boiling water, and exposure to sunlight to sterilize instruments and disinfect wounds. Sample types for testing. Before we move on to how we culture and identify microorganisms, let's first explore the type of samples we receive for testing. The microbiology lab handles a wide range of sample types. These include blood, used for blood culture, serological testing, and identification of bloodborne pathogens. This sample type helps diagnose infections such as bacteremia and sepsis. Urine, analyzed for urinary tract infections and sexually transmitted infections such as chlamydia. Stool, examined for gastrointestinal infections and parasites. Sputum/bronchial washing, tested for respiratory infections including pneumonia and tuberculosis. CSF, analyzed for meningitis, encephalitis, and other central nervous system infections. Corneal scrapings, primarily taken to diagnose infections of the cornea, also known as keratitis. Tissue biopsies, analyzed for identifying bacterial, fungal, or parasitic infections, as well as malignancies. Fluid samples, for instance, pleural fluid, acidic fluid, examined for infections and other pathological conditions. Hair, skin scrapings, and nail samples, tested for fungal infections such as tinea capitis. Swabs. The microbiology lab receives various swabs from different parts of the body. These include nasal swabs, taking for whooping cough diagnosis. Wound swabs, collected from infected wound sites to identify causative organisms. Genital swabs, used for detecting STIs such as HPV and gonorrhea, and genital tract infections such as balanitis in men and pelvic inflammatory disease in women. Throat swabs, collected for diagnosing bacterial and viral pharyngitis. Nasal swabs, analyzed for respiratory infections and nasal colonization by pathogens like Staph aureus. Ear swabs, collected for diagnosing otitis media and external ear infections. Eye swabs, used for diagnosing conjunctivitis and other eye infections. Skin swabs, analyzed for bacterial or fungal skin infections such as cellulitis and dermatophytosis. Vaginal swabs, collected for diagnosing vaginal infections such as BV and STIs. Rectal swabs, used for detecting gastrointestinal infections and colonization by pathogenic bacteria like Clostridium difficile. Some labs also receive environmental samples collected from water sources for monitoring microbial contamination and outbreaks. Food samples can also be tested for microbial contamination and foodborne pathogens. Specialized labs also receive animal samples to analyze for zoonotic infections and veterinary microbiology purposes. Handling culture medium. Now that we understand the importance of maintaining a sterile work environment, let's look into some of the ways we ensure the cultivation of our desired microorganisms with minimal risk of contamination. Aseptic technique. This is just a fancy term used to describe the practices we employ to prevent contamination and maintain the purity of cultures. In the lab, we achieve this by applying the following principles. Hand hygiene. Thoroughly washing our hands with soap and water before and after handling cultures. This also includes wearing the appropriate PPE, such as gloves, to further minimize the risk of contamination. Sterilizing equipment. Autoclaving glassware before reuse. Passing metal instruments through a flame to sterilize them before and after each use. Use of a Bunsen burner. Maintaining a flame in the vicinity of work areas to create a flow of air that keeps airborne contaminants away. Minimizing exposure. Keeping culture plates and sample lids closed when not in use to limit the time that they are exposed to the environment during manipulations. It's also a very good idea to create a designated separate space for use and clean equipment whilst working to prevent cross-contamination. Proper waste disposal. Making appropriate use of dispose-safe jars to safely dispose of biohazardous waste. Culture medium. We use culture media for the cultivation, isolation, and identification of microorganisms. This facilitates diagnosis and various other applications like pharmaceutical testing. Microbes like us have diverse preferences. To encourage the growth of specific types, we have different culture media options. These include selective media. Imagine selective media as an exclusive nightclub. Special ingredients in the media act like bouncers, allowing only specific microbes with the right, certain characteristics to thrive. This effectively excludes unwanted microbes, ensuring your target organism takes center stage. An example of this is McConkey agar. Differential media. Think of differential media like a costume party. It allows us to distinguish between different microbial types based on their reactions to the media's ingredients. Some microbes might turn the media a specific color, while others leave it unchanged. This color-coded response helps us easily identify the desired microbe. Mannitol salt agar is an example of this type of media. Enrichment media. Sometimes the microbe of interest is present but in low numbers. Enrichment media acts like a special energy drink for the shy guests. It provides them with additional nutrients they need to flourish, making them easier to detect and study. Selenite broth is an example of this. Sample inoculation and subculturing. Inoculation is the process in which a sample is introduced onto liquid or solid culture media to initiate growth of microorganisms. This medium provides the microbes with the nutrients they need to multiply and thrive. Techniques include streak plate method, pour plate method, and spread plate method. We choose the specific technique depending on the experiment and desired outcome. Subculturing involves transferring single colonies of the microbes of interest from their original culture to a new fresh medium. We often apply this process for a few reasons. Helps to isolate pure cultures for further testing, facilitates the preservation of cultures such as freezing, allows for larger quantities for further analysis, testing, or manipulation, for instance, sending to reference labs or performing susceptibility testing. Biochemical testing. Culture methods help us grow these culprits, but sometimes we need a little more information to get a definitive ID. That's where biochemical tests come in. Think of these tests like asking the microbes a series of quick questions about their unique abilities. Every microbe has its own set of talents. Just like people have different skills, microbes have different metabolic characteristics. Biochemical tests are designed to detect these unique talents and further aids in its identification. For example, a catalase test checks if a microbe can produce an enzyme that fizzes when exposed to hydrogen peroxide. Different microbes have different fizzing abilities, helping us differentiate them. Other tests include coagulase, indole, and oxidase testing. These tests are really important because they help us differentiate between similar species of microbes effectively, and because they provide results faster, they allow for quick diagnosis and treatment decisions. Microscopic identification of microorganisms. Microscopes are used in addition to biochemical testing to further aid in identification. Light microscopes use visible light to magnify a specimen and allows us to see basic shapes and sizes of microbes, often with the help of special staining techniques. Fluorescent microscopy, which is another form of light microscopy, is used to help identify fungal pathogens and acid-fast bacilli in clinical samples. Electron microscopes use electrons instead of light, revealing incredibly detailed structures of microbes at much higher magnifications. Electron microscopy is instrumental in visualizing viruses due to their small size, which is beyond the resolution of light microscopes. Under the microscope, a wet mount, also known as a wet prep, is used to examine living organisms in a fluid suspension. A wet prep involves placing a drop of liquid containing a sample on a slide, adding a cover slip, and examining it immediately under a microscope. It's a quick and easy way to gain preliminary insights into a sample without the need for staining or fixing. Stool samples are often examined this way to view parasite eggs and cysts that may be present. Iodine staining can be used to enhance parasitic structures to aid in identification. Blood smears are also examined under the microscope to detect blood-borne parasites like Plasmodium species, which causes malaria. To enhance the visibility of microbes under the microscope, we use some of the following staining methods. Gram staining. This is a common technique used to differentiate bacteria based on their cell wall composition. Gram-positive bacteria retain the crystal violet stain and appear purple under the microscope, while gram-negative bacteria appear pink after counterstaining with safranin. Bacteria can often be identified by their shape and arrangement, but further testing is required to identify the bacteria to species level. Fungal specimens can be examined under the microscope using potassium hydroxide preparation, which dissolves cellular material, revealing fungal structures such as hyphae, spores, and yeast cells. Both lactophenol cotton blue or calcofluor white are commonly used stains for fungi. They enhance the visibility of fungal structures, aiding in identification. Acid-fast staining. This technique targets microbes with a waxy outer coat, like Mycobacterium species, and some fungi. These microbes resist staining with most dyes, but this technique highlights them for better visualization. Did you know? Under fluorescent microscopy, fungi can resemble lightning or thunderstorms. Fungal hyphae, the branching filaments of fungi, can exhibit bright, intricate patterns that resemble the flashes, adding an artistic touch to scientific study. Serological procedures for common pathogens. Serological procedures are essential in diagnosing infectious disease by detecting specific antibodies or antigens in a patient's blood serum. Here's a quick recap of the definitions. Antigens are substances which elicit an immune response in the body. They include toxins and chemicals and often lead to the production of antibodies. Now, antibodies are proteins produced by the immune system to attack foreign bodies and fight off infections. While blood serum is the choice of sample to use for serological procedures, other sample types such as cerebral spinal fluid, urine sputum, and synovial fluid can also be used for testing. Here are some of the commonly used serological tests in the lab. Enzyme-linked immunosorbent assay, also known as ELISA. This test detects antibodies or antigens in a blood sample. It's widely used for diagnosing infections such as HIV, Lyme disease, and hepatitis. Rapid antigen tests. These tests quickly detect the presence of specific antigens, providing rapid results. They can be used to diagnose influenza and streptococcal infections. Serum agglutination tests. This test detects antibodies by causing visible clumping, which is known as agglutination, when mixed with antigen-coated particles. It is often used to diagnose infections such as brucellosis. Western blot, a confirmatory test that detects specific antigens in a sample. It's commonly used after an initial positive ELISA result to confirm Lyme disease and HIV infections. Susceptibility testing. Antimicrobial susceptibility testing is such an important tool in microbiology. Essentially, susceptibility testing is a laboratory procedure used to determine the effectiveness of antibiotics against specific bacteria and fungi in clinical samples. There are a few methods used in the lab. Disc diffusion method. Antibiotic discs are placed on agar plates with bacteria. Zones of inhibition indicate susceptibility or resistance. Minimum inhibitory concentration. This test determines lowest concentration of an antibiotic that inhibits bacterial growth. Various methods like broth dilution or e-tests are used. Automated systems. These systems use advanced technology to rapidly test multiple antibiotics against various bacterial strains, providing fast, standardized results. Molecular methods like PCR can detect specific resistant genes and pathogens, aiding in susceptibility testing. Effectiveness is assessed by comparing the result of these tests with established breakpoints, indicating whether a microbe microbe is susceptible, intermediate, or resistant to an antibiotic. This helps clinicians choose the most appropriate treatment. Not only does the procedure allow for effective treatment, but it also helps combat resistance by reducing misuse and aids in faster recovery and reduced risk of complications. This means shorter hospital stays for patients. Targeted dosage also means minimizing the risk of side effects. Fun fact: did you know that the discovery of penicillin was totally accidental? It all began with a moldy petri dish in Alexander Fleming's laboratory. He noticed that the mold Penicillium notatum was surrounded by a clear area containing bacterium Staphylococcus. He observed that wherever the mold grew, the bacteria failed to thrive. This chance observation led to the discovery of penicillin, the world's first antibiotic. Penicillin is still widely used today to treat bacterial infections. Well, folks, we have come to the end of episode 2. Join me again on the next episode where I'll be teaching you guys about infections based on different organ systems. Please make sure to like, comment, and subscribe. See you in the next one.